Yeah, welcome everybody to today's lecture about safety aspects of pluripotent stem cell-based cell and gene therapy. For the next 60 minutes, I would like to present to you some information about ATMPs, which stands for Advanced Therapeutic Medicinal Products. We will focus specifically on two specific groups of ATMPs, namely somatic cell therapy medicines and gene therapy medicines. These medicines address indications in the field of oncology, regenerative medicine, and genetic diseases. Now, what is the definition of somatic cell therapy medicinal products? SCTMPs, if you want to abbreviate it, contain or consist of cells or tissues that have been subjected to substantial manipulation such that the biological characteristics, physiological functions, or structural properties relevant to the intended clinical use have been altered. Or somatic cell therapy medicinal products consist of cells or tissues that are not intended to perform the same essential functions in the recipient and in the donor. Somatic cell therapy medicinal products are represented as having properties for or are used in or administered to human beings with a view to treating, preventing, or diagnosing a disease through the pharmacological, immunological, or metabolic action of its cells or tissues. Now, let me present to you some examples of somatic cell therapy products that are currently either involved in clinical studies or already applied on patients. So first of all, we have products that address cardiovascular regeneration, like hematopoietic or mesenchymal stromal cells for heart regeneration. A very famous example is represented by embryonic stem cell or induced pluripotent stem cell derived cardiac progenitor, cardiomyocytes for myocardial infection. There's a very famous example that was actually developed in Germany by the lab of Professor Zimmermann in Göttingen. And this product is termed heart patches, and they consist of engineered human myocardium for heart repair. Somatic cell therapy medicinal products can also be used for cartilage repair. And in this case, you have autologous chondrocyte transplantation, first, second, and third generation products. Somatic cell therapy products are used for skin regeneration, for example, skin cells that include keratinocytes and fibroblasts in combination with sheet-like matrices or scaffolds. We have skin regeneration for acute wounds, diabetic foot skin ulcers, and there's also products available against bullosa epidermolysis, chronic venous ulcers, peripheral arterial occlusive diseases. These are also products that are generated in or developed in Germany. They are derived from skin, ABCP5 positive mesenchymal stem cells, and the other product is expanded placental arterial stromal cells. There's also somatic cell therapy products to address acute graft versus host disease. This includes bone marrow derived mesenchymal stem cells, and in the case of loss of vision, there's a therapeutic approach available, which is based on limbal stem cells for limbal stem cell deficiency, embryonic stem cell derived retinal pigment epitheliums for age-related macular degeneration. And last but not least, there is a therapeutic approach available based on somatic cell therapy products, tackling complex perianal fistula or Crohn's disease, and this therapy is based on adipose tissue-derived mesenchymal stem cells. Now, what's the definition of gene therapy medicinal products? Now, GTMPs contain an active substance which contains or consists of a recombinant nucleic acid used or administered to human beings with a view to regulating, repairing, replacing, adding, or deleting a genetic sequence. Also, its therapeutic, prophylactic, or diagnostic effect is directly related to the recombinant nucleic acid sequence it contains, or to the product of the genetic expression of that sequence. Let me present to you one famous example of a gene therapy medicinal product that was approved in Germany since 2018. This is a CAR T-cell therapy to treat B-cell acute lymphoblastic leukemia. People that are eligible for this therapy are children and young adults up to 25 years of age whose cancer did not respond to previous treatment. has come back 2 or more times, or has come back after transplant of stem cells. So these are just examples of gene therapy products. And now we move on now to pluripotent stem cells. And let me first explain to you why in Germany the majority of products working with pluripotent stem cells are based on induced pluripotent stem cells. There's clearly a preference for iPSC work in Germany, and that results from the German so-called Stammzellgesetz, which is applied since July 2002. And this Stammzellgesetz makes people prefer to work with iPSCs. You see this written in German now. Let me just translate the 3 basic features that are critical if you want to work with human embryonic stem cells in Germany. You can work with an ESC line if the project includes high-ranking research goals for scientific knowledge gain in basic research or for the expansion of medical knowledge during the development of diagnostic, preventive, or therapeutic treatments for human applications. This is one criterion. The second criterion is you can use HESCs if there was a primary treatment of question to be addressed with the research project by applying in vitro models in animal cells or in vivo animal models. And last but not least, the third criterion is that HESCs can be used if the aspired knowledge gain can exclusively be accomplished by using embryonic stem cells. And these 3 criteria basically encourage many people who want to work with pluripotent stem cells in Germany to work with induced pluripotent stem cells because it's much less complicated, and in many cases the reviewers argue that most of the questions you want to address with ESCs can be addressed with iPSCs. Not all of them, but many of them. So this will be the reason why most of the data I will present now, information I provide, is dealing with iPSCs. Now, how about embryonic stem cell-based therapeutic approaches applied in clinical studies in Germany? The interesting thing is that the execution of clinical studies or research with human embryonic stem cell-derived intermediates or embryonic stem cell-derived differentiated somatic cell types is permitted. What is unclear is the regulation of the commercial utilization of embryonic stem cell-based therapeutic cells in Germany. This is still unclear because there are currently no products in Germany based on human embryonic stem cells. In the next slide, I would like to present potential applications of induced pluripotent stem cells. iPSCs are generated from healthy or diseased individuals by taking samples, for example, from the dermis, from the skin. These differentiated somatic cells are reprogrammed then as a next step by expressing a certain cocktail of reprogramming factors like Thomson factors, Yamanaka factors that are listed here, consisting of OCT4, SOX2, NANOG, or LIN28, or in the case of Yamanaka, OCT3, 4, SOX2, KLF4, and CMYC. And after a successful reprogramming process has occurred, you have pluripotent cells. If they come from a patient, they are patient-specific iPSCs. If they come from a healthy individual, they can also be used. These iPSCs are then differentiated into the cell type we are interested in, specialized differentiated cell types, and either the cells are then transplanted into the patient without any gene correction because the cells are healthy, or if the cells are diseased, have, for example, a genetic disorder or come from a patient with a genetic disorder, the genome is corrected, the gene is corrected, and the cells with a corrected genome are then transplanted into the patient. One risk that comes with iPSC-based cell therapeutic approaches is teratoma formation, which is based on huge amount of pluripotent stem cells that are introduced into the patient, but we'll get to this problem later during the talk. A huge advantage of using iPSCs for cell therapeutic purposes is that you basically avoid all the ethical issues that come with human embryonic stem cells, and autologous cells that are compatible with a patient's immune system can be used for therapy. Now, this slide basically summarizes the temporal distribution of clinical studies using embryonic stem cells and iPSCs, but also somatic cell nuclear transfer and parthenoid PSCs. This graph shows that until 2018, the majority of pluripotent stem cell-based therapeutic approaches were based on embryonic stem cells, but since 2018, the number of therapies that are based on iPSCs are basically skyrocketing. And right now we have internationally more iPSC-based clinical trials ongoing than embryonic stem cell-based clinical trials. What about the distribution of these trials internationally? You can see that the darker blue the color is, the more trials based on pluripotent stem cells are happening in a given country. You can easily see that of the 87 PSC-based clinical trials that were done since were done so far, the majority is happening in the US and in China, and this is even more demonstrated by this bar diagram that basically summarizes the sponsor type per country, and it shows you the number of clinical trials that were funded by different sponsors, for example, by academic government or by industry. Well, you can see that really of these 87 PSC-based clinical trials, again, the US and China is dominating, also Japan, and the European countries are not really characterized by huge numbers of clinical trials based on pluripotent stem cells. France and the UK has 2 trials so far ongoing. Germany is really bad with only 1 trial, as Sweden, and Switzerland also has only 1 trial ongoing. So there's a lot of room for improvement, and it would be really good if we would have more clinical trials based on pluripotent stem cells in Germany. So what kind of pluripotent stem cell derived cell types are involved in these clinical studies for the last decade? This heat map shows you that especially the clinical trials or pluripotent stem cell based clinical trials that generate retinal pigment epithelial cells or work with retinal pigment epithelial cells are dominating all the PSC studies. The next differentiated cell type that is based on pluripotent stem cells are natural killer cells, followed by mesenchymal stem cells, again followed by cardiac muscle cells. So these are the cell types in which pluripotent stem cells are differentiated into for therapeutic purposes right now. The induced pluripotent stem cell-based clinical trials that happen worldwide right now address different kinds of diseases, such diseases that have to do with mental behavior or neurodevelopment, diseases that are involved have to do with the visual system, with the respiratory system, with blood and blood-forming organs, with neoplasms, nervous system, circulatory system, endocrine and nutritional, metabolic diseases, diseases that have to do with the skin, genitourinary system, and developmental anomalies. So there are certain safety risks of human pluripotent stem cell-based therapies. The 4 risks, the major dominating risks, are summarized on this table. First of all, what is very intrinsic to pluripotent cells is that, as I already mentioned, undifferentiated human pluripotent stem cells can generate teratomas. Well, teratomas are benign germ cell tumors, and they form tumors when transplanted into immunodeficient mice. And although all clinical applications require cells to be differentiated, there's a concern about residual undifferentiated cells in transplanted populations. The big question here is always how many undifferentiated cells can be included in a cell therapeutic product before teratomas are formed. So there's a certain threshold value which cannot be really clearly defined and depends on the final product that is to be used for the patient. So the Food and Drug Administration in the US thinks that teratoma formation is a major obstacle to stem cell-based regenerative medicine. The second risk that is inflicted on patients treated with human pluripotent stem cell-based therapies is the common genomic changes that occur during generation and long-term culture of HPSCs. There are already older studies that showed that when you analyze more than 100 human pluripotent stem cell lines, that late passage age PSCs are twice as likely to have genomic changes than early passage cells. There's a selective pressure exerted over time in culture that may be a major contributor to genomic instability in embryonic stem cells induced pluripotent stem cells. Many of the same chromosomal abnormalities are found in both embryonic stem cells and induced pluripotent stem cells. Now, the third risk that is inflicted on these treatments are genomic mutations in transplanted cells that are associated with tumorigenicity. One should emphasize that every mutation causes cancer. So there are many mutations that just corrupt the efficiency or efficacy of cell types. But there are also those mutations, I should say the efficacy of a therapeutic cell type. But there are also mutations that are associated with tumorigenicity. And last but not least, there's the risk that the reprogramming method adds to the mutational load of cells. There are studies that show that reprogramming with different methods, mRNA, sendivirus-based or retrovirus-based, exhibit differences in the risk of causing mutations in the genome. And we now go more into the details of genetic and epigenetic abnormalities that are caused by reprogramming process and cultivation. The abnormalities, the genomic abnormalities that we find are karyotypic aberrations like aneuploidies and translocations, copy number variations, the acquisition of individual point mutations, and the loss of heterozygosity. The reprogramming process goes along with a massive global epigenetic remodeling, which includes histone alterations, de novo DNA methylation, and genome-wide demethylation. The second phenomenon that was observed is that the reprogramming process per se triggers the mobilization of endogenous mutagenic transposable elements. We will get back to this later. And thirdly, there are several reprogramming factors that were used and actually are still sometimes used are oncogenes. The most famous example are proteins of the MYC family, especially c-MYC. The first generation reprogramming factors included actually c-MYC, and this c-MYC is now more and more replaced by L-MYC, which is not an oncogene. While MYC was found to be not essential for IPC generation, its omission leads to a lowering of induction of efficiency by factor 100. They identified other factors shown to generate IPCs, revealed that they were linked to tumor formation. Now, the next issue is the issue of the reprogramming methods. And we mentioned this already briefly. I will get back to this in the next slide. And there's immunogenicity of allogeneic transplanted iPSC-derived cells. There's an immune response against iPSCs that is strong enough to cause rejection of cells. There are several strategies developed right now, for example, generating specific banks, haplotyped iPSC banks, that should reduce this risk. Now let's move more into details of the genetic and epigenetic abnormalities. So what you see here is a visualization on the right-hand side of duplications and other abnormalities that are observed frequently in iPSCs, and there are several chromosomes involved. Now, genetic instability in iPSCs typically results from the shift from oxidative respiration to oxidative glycolysis that is leading to an increased reactive oxidative stress. And the increased oxidative stress, leads to impaired DNA repair mutations, double-strand breaks. And this again is linked to genomic integrated reprogramming methods, oncogene reprogramming factors, particularly actually c-MYC. There are frequently in pluripotent stem cells observed non-random mutations on chromosome 1, 12, 17, 20, and chromosome X. Most of these changes are due to gains rather than loss of genetic material. Most aberrations are duplications. This is observed in the case of chromosome 12, 17, and X, and subchromosomal regions in pluripotent stem cells. Commonly found duplications include genes that give cells a growth or survival advantage. And as the cells divide in culture, there's selection for those that are better adapted to culture conditions. There's also frequent amplification of a specific region of chromosome 20, as shown here on this graph. And last but not least, what is frequently observed are single nucleotide variants. They are point mutations in coding and non-coding gene sequences. So let's move on to the second factor that can destabilize the genome, and this is the observation that the reprogramming triggers the mobilization of endogenous transposable elements. It was shown several years ago that endogenous transposable elements that are normally silenced in differentiated cells, as shown here, they are methylated in their promoter region, and this methylation ensures that these elements are silent and not transcribed. However, if now reprogramming is initiated by adding Yamanaka or Thompson factors, there's a global demethylation initiated in these cells, and the consequence is that these endogenous functional line elements, of which we have about 150 in our genome, are transcriptionally activated. The activation induces expression. The expression of those functional L1 proteins induce a retrotransposition cycle and new insertions of those transposable elements. And the consequence is not only mobilization of line 1s, but also of ALU and SVA elements that occur in iPSCs. This actually has also been observed in continuously pluripotent cells like embryonic stem cells. Interestingly, the majority of those insertions was observed to occur into introns of host genes. And as I will show you later, this will also affect host gene expression. So taken together, these data showed, and this was already reported in 2016, that reprogramming triggered mobilization of endogenous L1, ALU, and SVA elements occurs and can add to the corruption of genomic integrity and biosafety of iPSCs and their differentiated derivatives. So let's now talk about the reprogramming method affecting the mutational load in pluripotent stem cells. What you see here on the left-hand side is a schematic of experiments where they cause the derivation of 3 biological replicates of each of 3 reprogramming methods: mRNA-based reprogramming, retrovirus-based reprogramming, and Sendai virus-mediated reprogramming. And what was found is shown here on this bar diagram, where you see the number of high confidence variants indicated as bars. And it was shown, if we sum up the data that are presented here, in the case of retrovirus-induced cell lines, that those harbored more potentially damaging mutations than the other methods. And also, these cell lines contained integrated vectors in 7, 8, and 12 genomic sites, which of course can cause insertion mutagenesis or also, worst case scenario, activate proto-oncogenes. The majority of integration sites were intergenic, some integrations mapped to coding regions. So what happened with, in the case of the reprogramming with mRNA, there's no integration of the mRNA possible. Nevertheless, several variants that overlapped the binding sites for transcription factor showed mutations. Furthermore, if you look at Sendai virus reprogrammed cell lines, again, in the case of Sendai virus, there's no integration observed. Sendai virus do not integrate normally. Those cells have a trend towards fear-coding mutations, which means mutations in coding regions. So if we summarize these findings, of where did we find all those mutations observed caused by the reprogramming method, most mutations fell with intergenic or intronic regions. The rate of coding mutations were 2 to 10 mutations per cell line. All of the iPSC lines had hundreds of variants compared with parental fibroblasts, the mRNA-derived lines having least high confidence mutations on average. And to make a long story short, they identified trends that linked reprogramming strategies to certain mutations. Taken together, one of the safest methods to reprogram differentiated cells into pluripotent stem cells are mRNA-based and Sendai virus-based reprogramming methods. So now let's move on to gene therapeutic approaches that are based on pluripotent stem cells. What is gene therapy? And this is a definition that was presented by the National Cancer Institute. It's an experimental treatment that adds a new gene or replaces or repairs a mutated changed gene inside the body's help to help prevent or treat certain diseases, such as cancer. Gene therapy may also be used to train the body's immune system to recognize and attack cancer cells or to protect healthy cells from the effects of cancer treatment. It's being studied in the treatment of some types of cancer. But of course, gene therapy has also the potential to treat other diseases than cancers, like genetic diseases. So what's the definition of genotoxicity? Genotoxicity is a collective term that includes any process which affects the integrity of genomic DNA. A big problem is genotoxicity in gene therapy, and what is meant here is the insertion of genetic elements, for example vectors, and the expression of DNA-modifying proteins that are the main mediators of genotoxic side effects. So if you use vectors for gene therapy, the most frequently used vectors are shown here in this little box. Adenoviruses, for example, which is the most widely used vectors in gene therapy with more than 500 clinical assays. Retroviruses are used, lentiviruses, adeno-associated virus, herpes simplex virus, and non-viral vectors. And I marked those lentiviruses and AAVs in red because I will show you a couple of examples for lentiviral adeno-associated virus-based pluripotent stem cell-based gene therapies. Here's an example for a CAR T-cell immunotherapy against solid tumors based on iPSCs that were transduced with a lentiviral vector before. And what happened here is a deriving and expanding of CAR T-cells from a single iPSC clone. This is a brand new therapeutic, a clinical trial is going on based on this iPSC-based CAR T-cell line. A second example is the transduction of iPSCs with adeno-associated virus. And again here, the development and the testing of gene therapy for neuromuscular and motor neuron disorders was based on an AAV viral gene delivery or transduction. So what are the safety concerns that are associated with viral vectors, which include those that can lead to genotoxic events? For example, random insertion can disrupt normal genes. There's activation of proto-oncogenes. There's insertion mutagenesis. And last but not least, viral vectors introduced in the cells can cause inflammation. Some of these problems that are caused by viral vectors in gene therapy can be avoided by using instead genome editing techniques. These genome editing techniques address certain limitations and reduce particular risks of genotoxicity in viral vector-based gene therapy. What's the definition of gene editing? Gene editing is the ability to make highly specific changes in the genome, applying technologies that allow genetic material to be added, removed, or altered at particular locations in the genome. Gene editing is performed using enzymes, particularly nucleases that have been engineered to target a specific DNA sequence, where they introduce cuts into the DNA strands, enabling the removal of the existing DNA and the insertion of replacement DNA. There are different approaches of gene editing. There are basically two specific approaches. There's a gene knockout approach and a gene knock-in approach. Both approaches rely on generation of DNA strand break by a nuclease. And of course, the response of the cell always is it tries to repair the break either via non-homologous end joining repair or homology-directed repair. These are the two repair mechanisms that you find in human cells. The repair response to the breakage defines the resulting gene editing approach. Let's now zoom into the gene knockout approach. Why are we doing gene knockouts? To understand function of gene of interest, the first gene editing approach that everybody performs is to render the gene non-functional and to study the consequences of this knockout. And the gene knockout always relies on non-homologous end joining repair. The gene knock-in can be, or the general procedure of gene knock-in can be divided in three different kinds of knock-ins. They serve different purposes. The insertion of a transgene into the defined locus means that a gene is added to the targeted cell and its function or effect on the phenotype can then be explored. This can be done by knock-in experiments. The insertion of a small or large tag into the coding sequence of a gene is the second possibility. What you do is you basically add a sequence to facilitate the detection or the purification of the gene product for biochemical assays. And the third strategy is to generate a point mutation by exchanging base pairs. This is mainly used to model and eventually treat the effect of point mutations in the context of genetic diseases based on homology-directed repair. This is frequently done right now already also with the help of pluripotent stem cell-based approaches. Now let's talk about the three different major groups of gene editing. In 2008, the first successful gene editing approach was based on zinc finger nucleases. Using zinc finger nucleases, the principle of gene editing was demonstrated successfully for the first time. Zinc fingers contain a nuclease domain and specific zinc finger DNA binding domains that recognize 3 base pair sites on DNA, as shown here. Multiple zinc fingers can now combine to form dimers. The dimerization process permits to recognize longer nucleotide sequences, amplify the specificity, and improve the adaptation to the target. The second gene editing approach is based on transcription activator-like effector nucleases, or abbreviated TALENs. This strategy was developed in 2011. TALENs brought the recognition down to the single nucleotide resolution, and they also contain a nuclease fused to DNA binding domain sequence, but they are capable of single nucleotide recognition, thereby increasing targeting capabilities and specificity compared to zinc finger nucleases. But there are major drawbacks of both platforms, zinc fingers and TALENs. And this is the requirement of designing and validating a new zinc finger nuclease or TALEN protein for each new target editing site. Now, the third group of gene editing strategies is based on CRISPR-Cas mediated precise genome editing systems. And these are shown on this slide as a summary. The traditional CRISPR-Cas mediated homology-directed repair was developed in 2013 and was already used successfully, frequently, several times already. The base editing approach, which is also based on CRISPR-Cas, was developed in 2017, and then there's the third approach, which is termed prime editing, was developed 2019. I will just in a second get to the reason why CRISPR-Cas mediated HDR was further developed into these other approaches, or what the advantages and disadvantages of these approaches are. But what they all have in common is basically the CRISPR technology. So CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. The CRISPR-Cas technology is based on a guide RNA and the Cas9 endonuclease, and the guide RNA recognizes the target site, and the Cas9 endonuclease cuts and triggers the repair mechanism. Basically, all these three approaches, CRISPR-Cas mediated HDR, base editing, prime editing, are based on the original CRISPR-Cas technology. So let's zoom into a little bit more into the CRISPR-Cas mediated homology-directed repair. Cas9, and this is shown in this image here, nuclease is guided by a single guide RNA to generate a target DNA double-strand break in the presence of a repair template carrying the desired edits, and this is shown in orange here. The cellular homology-directed repair machinery repairs the double-strand break using the template, allowing precise incorporation of the template carried edits in the genome. Now, the big problem of all these editing effects are off-target effects, and the CRISPR-Cas9 system's off-target effects mainly result from the generation of DNA double-strand breaks. Whenever you generate a DNA double-strand break, it's bad for the genome and recombination cannot occur, or the repair doesn't work perfectly. So these double-strand breaks are repaired at target genomic loci, and the requirement to include a donor DNA molecule and the activation of DNA repair mechanisms then cause off-target effects that could damage the DNA and the DNA integrity. Double-strand breaks are always repaired by homology-directed repair or non-homologous end joining. NHEJ, non-homologous end joining, is error-prone and generates insertions or deletions. So the own host-encoded repair mechanism is responsible for insertions or deletions that are defined as off-target effects by CRISPR-Cas. So the second approach that is based on CRISPR-Cas technology is base editing. Base editors are engineered fusion proteins that are composed of a almost dead Cas9 protein. It has a mutation in a D10A mutation and deaminase domains. Cytosine base editors can convert cytosine to uridine in a single strand. The resulting UG heteroduplex can then be converted to a TA base, as it is shown here, following DNA replication or repair. Another functional domain fused to the cytosine base editor, the uracilglycosylase inhibitor UGI domain, prevents uridine from reverting to C, thereby favoring the C to T conversion. Adenine base editors can deaminate, and this is shown here on this part, can deaminate adenosine to form inosine, and that has the same base pairing preferences as guanosine. Thus, the IT heteroduplex can be converted to GC following DNA replication and repair. Both base editors, cytosine base editor and adenine base editor, in both cases, the modified Cas9 protein nicks the target strand to create single-strand breaks and not double-strand breaks, as in the case of CRISPR-Cas-mediated repair. The mismatched repair machinery then pairs the deaminase converted base with a properly matched base. Now, the crucial part with base editing and also prime editing is that they only cause single-strand breaks, and this avoids the majority of off-target effects, but not entirely. Now let's therefore then focus now on prime editing. The prime editor is an engineered fusion, that is shown here on this part of the slide. The prime editor is an engineered fusion protein composed of nickase, the NCas9 that is also used for base editing, and reverse transcriptase. NCas9 creates a single-strand break on the non-target strand. The release 3' end is then hybridized to the 3' end of the prime editing guide, the pegRNA, and is reverse transcribed by the RT domain. The reverse transcription process incorporates the edits encoded in the pegRNA into the newly synthesized DNA strand, an equilibration between the edited 3' flap and the unedited 5' flap. Endogenous 5' flap cleavage and ligation and DNA repair results in the stable incorporation of the desired edit in the genome. So what's the big difference between prime editing versus base editing? Prime editing is more versatile because it can precisely perform targeted small insertions, deletions, and base exchanges while limiting negative effects. With regard to off-target effects, prime editing is the way to go. What sets those tools apart from the CRISPR-Cas9 technology? Well, it's basically the ability to make targeted edits without breaking the double strand. The prime editing technology is built with a Cas9 nickase, which induces single-stranded breaks in DNA, fused to reverse transcriptase enzyme. It uses a single engineered construct known as a primed editing guide RNA, which is made up of the primer binding site sequence and a sequence containing the desired edit. When it comes to cell therapeutic products that are used for clinical trials, there are more and more products coming up and provided to the national authorities that are based on prime editing. And this brings me actually to my gradient figure, where you basically see a gradient of the number of off-target effects generated by the different gene editing methods. The most off-target effects are generated by zinc finger nuclease mediated gene editing, and the lowest amount of off-target effects are observed in the case of prime editing. So iPSCs derived from patients of a variety of diseases have been edited to correct diseases-associated mutations and to generate isogenic cell lines. After the differentiation process, many of the corrected iPSCs showed restored functionality and demonstrated their potential in cell replacement therapy. And I want to provide one example. There are many examples, but I want to focus on one right now, and this is the gene correction in the case of patients with cystic fibrosis. Now, cystic fibrosis is a condition that is formed by mucus in the lungs, impairing breathing and often leading to premature respiratory failure. The primary genetic defect in the cystic fibrosis transmembrane conductance regulator gene, or abbreviated CFTR, is the reason for this disease. CFTR encodes chloride channel protein, encodes the chloride channel protein that regulates lung epithelial chloride transport. Now, if mutations are introduced in the CFTR gene, these mutations prevent expression or reduce functionality of this chloride transporter at the cell membrane. So, what was done to correct this genetic disorder with the help of iPSCs? Two approaches were so far presented. In 2015, they were published almost at the same time. There was a combination of CRISPR and piggyback transposase technology to achieve footprint-free gene correction at the CFTR locus in patient-specific iPSCs. After this mutation was corrected, the cells were subsequently differentiated into lung epithelial cells, and these epithelial cells demonstrated recovered function and expression of CFTR. In parallel, at the same time, zinc finger nuclease technology was used to achieve gene correction of the same CFTR gene. And again, the corrected cystic fibrosis iPSCs were differentiated into lung epithelial cells. And also in this case, the mature CFTR glycoprotein was expressed in the restored cells, and the CFTR chloride channel function worked pretty well. Now, what are the methods to assess genomic integrity of pluripotent stem cells and their derived differentiated cell types? Now, you have to distinguish here if you want to determine the genomic integrity of the iPSC line that you want to differentiate, or if you want to determine the genomic integrity of the final differentiated cell type that you finally want to introduce into the patient. If you want to determine genomic integrity of the pluripotent stem line, in many cases, people want to generate working cell banks or master cell banks of those iPSC lines and then differentiate them in different cell types. Now, to characterize the integrity of pluripotent stem cells, no matter if iPSCs or embryonic stem cells, there are methods that are used to detect already known abnormalities. What I mean by that is these are abnormalities that were reported earlier in many iPSC lines. So these specific mutations are observed again and are recurring. And there are specific methods to detect those, and this is, for example, fluorescent in situ hybridization, which you use for the detection of whole chromosome abnormalities. There are single nucleotide polymorphism arrays or SNP arrays, where you detect, as the name says, single nucleotide polymorphisms. And there's digital droplet PCR, a highly sensitive PCR method that is used for single base resolution. And digital droplet PCR can also be used for high precision, in high precision for CNV, copy number variations, and single nucleotide variation detection. And last but not least, there's targeted sequencing. Targeted sequencing means you sequence only certain genes or regions in the genome that are crucial or that you want to specifically analyze in greater detail. For example, oncogene panels. These oncogene panels are changing every year because more and more oncogenes are detected, or genes that play a role in oncogenesis, and then these specific genes are sequenced to test the genomes for potential mutations, point mutations. So digital droplet PCR and the targeted sequencing both rely on a priori knowledge of the existence of recurrent mutations for primer design. You have to know where the mutations are expected to occur in order to design primers for these approaches. and for the analysis of the massive amount of identified DNA variants. The accumulation of info on recurrent iPSC genomic abnormalities and on mutations with phenotypic consequences is crucial, and that's why sponsors that develop products and sequence their pluripotent stem cells for abnormalities should provide those data to the public so that other laboratories or sponsors can use those data in order to increase the safety of the final product. Let me just briefly show you again my little gradient figure here on the right-hand side again. This gradient shows an increasing measure for precision. So FISH has a lower resolution than digital PCR, which you can use to detect single nucleotide variations. I should also emphasize that looking for already known or earlier reported PSC-specific genomic abnormalities is based on the so-called streetlight effect. That basically means, you have a street light that always illuminates only a certain region on the street and doesn't look at the dark parts of the region. You don't know what's happening in the dark parts of the street where the street light is not illuminating the street. So this first set of experiments or approaches is different from the methods covering the entire genome. Methods that cover the entire genome, where you can also detect abnormalities that you would not expect, is G-banded karyotyping, where you analyze whole chromosome abnormalities. There's array comparative genome hybridization, or abbreviated ACGH, and there is next generation sequencing, which again is used for single base resolution. To make a long story short, the advantage of next generation sequencing, and I'm actually including here short read sequencing and long read sequencing, have the advantage that you assess the whole genome at a single base resolution, and you can detect most genomic abnormalities with this method depending on your sequencing depth. The disadvantage so far was that the high depth sequencing is not routinely available due to cost, data analysis workload, and complex result interpretation. However, in the recent years, the costs for high depth sequencing are going down more and more, and it becomes more and more standard to laboratories to do deep sequencing. So you can actually, people do routinely sequencing depth of 65-fold now, which detects also small subpopulation of cells with mutations. What I also would like to emphasize are methods to identify double-strand breaks that are generated by Cas9 in vitro or in vivo, since the CRISPR-Cas9 methodology is now taking off and people are using it frequently. Also, the methods to identify the off-targets effect, there are more and more tools coming up to detect those effects. There are so-called digested genome sequencing. There is the genome-wide unbiased identification of double-strand breaks enabled by sequencing, or called guide sequencing. A brief schematic of this method is shown here on the right-hand side. You basically generate sequencing libraries of regions where the double-strand break occurred, and then you can detect where the off-target effects of a specific CRISPR-Cas approach is causing a problem. So then you can qualify your CRISPR-Cas approach before you apply it. You can qualify it for safety. So you can test if it has many off-target effects. There's a third method that is called high-throughput genomic translocation sequencing, and BLES, that stands for labeling enrichments on streptavidin and next-generation sequencing. All those methods are based on libraries of sites with off-target effects with double-strand breaks. So the take-home message of these methods that are available to characterize the genomic integrity in the case of applying pluripotent stem cells for therapeutic products is, as soon as the costs become acceptable for routine screening and as soon as there are robust analysis pipelines available, next-generation sequencing should become the favorite technique for iPSC genome integrity screening. And here I'm emphasizing not only short read but also long read sequencing, because with long read sequencing you can detect regions that are repetitive and are not detected by short read next generation sequencing. So here is some information on the sensitivity of different methods detecting genomic variants. So the abnormal clones representing only a fraction of total cell population could go undetected if this fraction of abnormal clones is small enough. These subpopulations pose a risk for the cell line quality because of the clonal expansion they can go through. So here are three methods presented that have different degrees of sensitivity. So for example, digital droplet PCR has a sensitivity that detects variants that are present in 10% of the iPSC population. FISH is already more sensitive. You can detect variants that are present in 5% of the iPSC population. But the winner is basically ultra-deep sequencing, where you can detect variants that are present in only 0.1% of the population. It should be emphasized that HIPC lines without abnormality at the derivation, when you generate a clonal iPSC line, could be mostly genetically abnormal already 5 passages later. So, therefore, certain recommendations for the clinical use of pluripotent stem cells were announced. The genomic integrity should be monitored around every 36 cell divisions after the iPSC line was generated. The standardized guidelines are currently not available. There's a recommendation by the EMA, a so-called reflection paper on stem cell-based medicinal products, which defines those recommendations. So from a regulatory perspective, the genome integrity monitoring pluripotent stem cells, specifically induced pluripotent stem cells that are less stable than embryonic stem cells, should be a mandatory quality control before their use in clinical trials. And here we go back to the 5 passages again. The quality control must be done shortly before the use, because an advantageous mutation can take over a cell culture in less than 5 passages. Ideally, the genome of clinical trials iPSCs, so iPSCs whose derivatives are used for clinical trials, should be analyzed less than 5 passages before the differentiation in therapeutic cell type to exclude at least the most recurrent genomic abnormalities. And this is actually what regulatory authorities demand most of the cases. Now, let me focus now more on the reprogramming issue, on the issue that reprogramming triggers mobilization of mutagenic endogenous transposable elements. As you know, only a small fraction, namely about 1.5% of the host genome, consists of exons of functional host genes. A huge fraction of the genome is the consequence of the activity of different kinds of transposable elements. There are, for example, the DNA transposons, which are fossils. They are not able to transpose anymore because they have too many mutations. There are endogenous retroviruses, which also cannot spread in the genome anymore, but some of them still produce proteins. But there's a huge fraction of non-LTR retrotransposons which is actually able to retotranspose, and it consists of 4 different subfamilies that are actively mobilized in human genomes under certain conditions. These families are short interspersed nuclear elements, or SINEs, that cover 13%, long interspersed nuclear elements, the lines, that cover about 21%, and there are the SVA elements covering 0.2%, processed pseudogenes covering 0.5%. On the right-hand side of the slide, you see summary of the structures of the different transposable elements in the genome. The line 1 element is basically termed the mother of the functional line 1 retrotransposons because it's autonomous, and it codes for the protein machinery that is responsible for the mobilization of itself, but also for the mobilization of the groups of non-autonomous non-LTR retrotransposons. The line elements are 6 kb long, code for 2 functional proteins, and mobilize ALUs, which are 280 base pairs long, SVAs covering 700 base pairs to 4 kb, and processed pseudogenes, which can vary in their length quite a bit. The functional line 1 retrotransposon is harboring 3 open reading frames. The smallest open reading frame is termed ORF0. which covers 216 base pairs. It's coding for 71 amino acid transacting peptide, which enhances retrotransposition. The second open reading frame covers 1 kb. It encodes a trimeric RNA binding protein, which can be subdivided in 3 domains, called call domain, RNA recognition motif, and a C-terminal domain. ORF1 is separated from ORF2 by a 63 base pairs non-coding spacer region. ORF2 covers about 3.8 kb. It encodes on the N-terminal end, or well, I should say on the 5' end and endonuclease domain, followed by a reverse transcriptase domain, and a cysteine-rich motif, which is essential for mobilization, but nobody really knows what it's doing. If you introduce point mutations, the element cannot retotranspose anymore. All full-length line, all retrotransposon insertions in genome are flanked by variable target site duplications. They have a 5' UTR. Line 1 elements have a 5' UTR with a gene internal promoter and a 3' UTR where transcription termination is initiated. The poly-A has a 3' end range from 14 to 40 adenolic acid nucleotides. And those elements, line elements, cover about 70% of the human genome. However, of the 10 to the 6 copies in our genome, 95% of the copies do not look like this, what you see here. 95% are 5' truncated or internally rearranged and therefore not functional anymore. But we still have 5,000 to 6,000 copies which look exactly like this figure shown here. Most of these copies again are inactive due to point mutations. That leaves 80 to 150 copies of these elements in the genome that have intact functional open reading frames and can mobilize in certain cell types under certain developmental conditions. They are termed retrotransposition competent. So what you see here is a brief schematic of the line 1 retrotransposition cycle. The entire slide is a human cell, and the region in the middle is this nucleus. And what you see here is one chromosome with one of the 80 to 150 functional L1 loci. This one locus is transcriptionally expressed, and the RNA is transported into the cytoplasm, where translation occurs, and the resulting proteins, ORF1P and ORF2P, these are the 2 proteins that are encoded by L1, have a cis preference for their own RNA, which means they bind to their own RNA and form a ribonucleoprotein particle consisting of many ORF1 proteins and only 1 or 2 ORF2 proteins. And this particle now moves back into the nucleus to the genomic DNA, where it initiates reverse transcription and integration of this new copy into the genome. I should say that 95% of all copies are 5' truncated, not functional. 5% are full length, and most of them then are also functional. Now, while L1 proteins have a cis preference for their own protein, in exceptional cases they can also mobilize with their proteins. L1 proteins can also mobilize other mRNAs like SVA RNAs or ALU RNAs and cellular mRNAs that are encoded that are expressed from RNA polymerase II, which are regular transcripts from regular host genes that are protein coding. But this happens rarely, and also those retrotransposition events are integrated into the genome. Transposition normally occurs in the germline, in precursors of gametes, and in gametes themselves. Furthermore, they occur in pluripotent embryonic stem cells, up to a certain step in embryonic development. Further somatic events are happening in tumors and in neuronal precursor cells. And in this slide, I just try to summarize the different mechanisms by which the activity of those elements can actually cause damage in a human cell. Line 1 retrotransposition, if it integrates into the genome, I think that's easy to understand, can cause insertion mutagenesis and interrupt genes. But line 1 activity does not necessarily have to integrate the entire element to affect integrity. It can also damage the genomic DNA by the endonuclease activity, by just nicking and cleaving the genomic DNA, as it happens in many tumors where line elements are highly expressed. And last but not least, line 1 mediated disease causing mechanism is the activation of the cGAS-STING pathway by activating innate immunity via reverse transcribing cellular mRNA into cDNA, and this cDNA is recognized as foreign, like a viral infection, and then initiating innate immune response. Now, as I already showed you this slide, our lab could a while ago already demonstrate that these elements are indeed activated in pluripotent stem cells, not only in embryonic stem cells, where this event, this transposition is happening frequently, continuously, but in iPSCs initiated by the reprogramming process, and then the mobilization happens quite efficiently, actually. And we could basically find at the time that the reprogramming triggered mobilization of endogenous L1, R1, and SVAs occurs and can add to the corruption of genomic integrity and biosafety of iPSCs and their differentiated derivatives. And I just want to show you a couple of examples that nicely show that the iPSC integrity can be affected by transposition events. What you see here are 11 transposition events observed in 2 or 3 different iPSC lines. The red marked boxes represent the insertion. The little white red bars indicate reads that just demonstrated the insertion in the iPSC line. And I just want you now to focus on those 6 loci that integrated into genes. For example, this is summarized here, 6 of those insertions occurred into genes, actually always in introns. And there were genes like, the target genes were SLAC12A1, which is an ion transporter that is highly expressed in metionephrons. Another gene was PTPN9, tyrosine phosphatase, that is active in oncogenesis. There was NREP, a neuronal regeneration-related protein. And this is a very interesting guy, RNF38, which is a ring finger protein 38 that plays a role in oncogenesis and apoptosis. Another insertion occurred in the plexine domain containing 2 that is associated with Sotos syndrome. And last but not least, there's this red-marked insertion in the CATPS2 gene, which is a calcium-dependent secretion activator 2. Now, this insertion is marked in red because we showed for this insertion that it really affects the expression of those genes. This insertion, although integrated only in an intron, and this was shown by generating single-cell clones from the population of iPSCs that harbor the insertion. As I said, not every cell has this insertion, only a subset of cells has the insertion. So we basically did single-cell cloning and identified those cells that have the insertion, those that don't have the insertion, and we isolated cytoplasmic RNA, did QRT-PCR, and then you can see that those cells that have the insertion in the intron have a dramatically reduced expression of the CATPS2 gene relative to cells that don't have the insertion. So clearly the CATPS2 expression is downregulated in the single-cell clone harboring this specific insertion in the intron. This is important because most of the insertions happen in introns and can thereby affect transcription levels of host genes. This next slide just shows that this phenomenon observed in human cells was also recently demonstrated to be true for mouse stem cells, where a collaborator of our lab showed applied short-read or Oxford nanopore technology long-read genome sequencing to 38 bulk murine iPSC lines that are reprogrammed from 10 parental cell types and to 18 single-cell murine iPSC clones, and they found 83 de novo transposable element insertions in those iPSCs. Interestingly, those insertions included insertions into the BRCA1 gene and DMD gene, and they also found that these radiotransposons in those iPSCs are profoundly hypomethylated in MIPSCs, which is consistent with what we found. And with this, I would like to finish my presentation, and thank you very much for your attention.